Okay, everyone, I'm going to get started. I am following Molly, and the two of us kind of decided to tag team because she spoke to you about gene editing and modulation in cells, and I'm going to talk to you about doing that same thing, but in mice. So I run the Mouse Genetics Core facility at National Jewish, and today I'd like to introduce you to the facility if you're not already aware of what we do, and give you some concrete examples of some of the projects that we've been working on. So the way I've organized my presentation, first I'll give you some background into the general types of services we offer, the culture of collaboration that we come into projects with, some examples of models we've made with classic methodologies, and then spend most of my time talking about how we are using CRISPR-Cas editing in mice to make knockouts and knock-ins with an unprecedented speed, as well as leveraging in vitro fertilization and CRISPR to work in generating mice on novel, different background strains or already genetically modified mice. And at the end, I'll give you a screen with my email address and how to best reach us. So my facility has been around for quite a while. I started this facility about the same time Molly did, about just over a decade ago. And originally we supported only the investigators at National Jewish. But over time, kind of organically, we started to take on more and more projects at the University of Colorado. And in 2018, we became your designated core facility as well. In addition to this, we support a lot of external investigators, including some from UCSF predominantly and La Jolla Institute. We are a fee-for-service facility, and all of our charges reflect the cost that we incur to provide those services through the purchase of reagents. At the bottom, I just wanted to illustrate how much our facility's grown over time, just to show you how many novel mouse models we create annually. And over the last 10 or 12 years, it's become an exponential service that we provide. We're comprised of six. We have 6 people in my group. In addition to me, I have some very talented scientists. Jimmy Gross came to me from Duke and Washington University in St. Louis. He's a microinjectionist who's been doing this for over 20 years. Anil Jain came to the core more recently after finishing his postdoc at the Anschutz Medical Campus. He's being trained to be a microinjectionist and is doing complex colony management and some phenotyping and genotyping of new models. Cindy is from CU Boulder, where she was doing some of the things that we do in our core here, so it was a good fit. Christina is an embryonic stem cell specialist and does embryo and sperm cryopreservation for mouse models. And Bi-Chen was a postdoc here at National Jewish, and I was lucky to acquire him when he finished his postdoc, and he is our in-house molecular biologist. So over the course of the last decade or so, we've expanded our facilities services, basically with the idea in mind that we want to be a full service facility and provide you complete support if you don't have any interest or skills devoted to developing new models and you just want to work on the mouse models. But we also work with investigators. So if you have an interest in working on some of these aspects, you want to develop your own constructs, for example, that would be absolutely fine. So here I've just outlined at very broad strokes the kind of services we offer. As I said, Bi-Chen does most of the molecular biology. This involves backward combineering, complex construct generation, such as embryonic stem cell targeting constructs. Christina does the embryonic stem cell targeting. Several of the people in my group do the microinjection, the introduction of these modifications into mouse embryos and transfer into females so that we can produce live animals. We have expanded our facility services to allow us to do the breeding to establish a germline transmission, genotyping. In some instances, we also generate cohorts and do preliminary analysis for investigators. And finally, once you've generated those animals, we will help you cryopreserve sperm or embryos from them if you want to close down a line, or we will import cryopreserved materials to help rederive live mouse colonies for your research. In addition to that, I do a little bit more. I also help identify the best mouse models for research questions that you might have. This includes generating letters of support, cost estimates, and help writing parts of grant proposals. And once we've made a mouse for you, or even if we haven't, and you're having problems with the mouse that you have, you're welcome to reach out to us as a resource. We can help you help provide you with literature, help you understand caveats and limitations of models, provide summaries of animal models that we've created for you, troubleshoot, and help with manuscript preparation. I kind of look at this as we're all in this together. I don't expect you to be an expert at mouse genetics, but I expect that when you work with us, you get the knowledge that you need to speak effectively about the models that you're working with and understand the strengths and limitations of those models. So these are just a few visual examples of some of the things that we do share with investigators when they start a new project with us, if they're doing a CRISPR-Cas project. So we do have tutorials that we've written up that just summarize the strengths and weaknesses of CRISPR-Cas in animal models. I often provide investigators with papers outlining some of the strengths and weaknesses, such as undesired bystander mutations that might be observed, off-target effects that might be identified, as well as how to best validate those new animal models. In addition, when we finish and we generate a new mouse model for you, we started to make reports for investigators. This is just a snip of the first page of that. But in essence, what we try to do is consolidate all of the information related to the projects that we made for you, with the idea that when you later go to publish that mouse a couple of years later, you have everything you need in this document to write up that materials and methods section. And it's a good way for us to keep track of everything that was done on your project over a period of time. But how are we different from your previous course? So it's become obvious to me as I've talked to investigators over the last few years that there are some things that are different. The main thing being, I think, the IACUC. We have our own distinct and completely autonomous animal protocol. So if you create a new animal with us, a new model, you don't need to add any amendments to your IACUC protocol for anything that we do for you. You would basically add a new strain to your protocol as if you're purchasing them from a vendor. So hopefully this facilitates your ability to work with these new models. In addition, for animal transfers in quarantine, both animal programs at the university and at National Jewish have IACUC approved transport, and we can receive or import animals for you for acute use, whether it's for sperm or embryo cryo or for IVF to generate modified mice on a modified background. We can import those mice and use them rather readily. And when we do end up transferring modified mice to you, they will go through quarantine, and I have it, Jory shared with me that the timing would be very comparable to what you would have experienced with your former transgenic and knockout core facility at the university. So I'd like to give you a couple of very brief examples of mice that we've made before CRISPR-Cas9 and other engineered nucleases came on the scene. Just to give you an idea, these are things that we still provide, but less commonly so, just because CRISPR-Cas has been such a powerful and transformative tool to make genetically modified mice. So we have generated a lot of mice using embryonic stem cell targeting to make knockouts and knock-ins and back transgenic mice. And we've done a little bit more unusual projects like tetraparental aggregation chimers and embryo transfers between strains, which I'm not going to go into today. This embryonic stem cell targeted mice is an example from John Cambier's group. His favorite molecule for a while has been STIN, which is a sensor for bacterial and anyway, so STIN is important to him. So he reached out to us and we've made him 3 different mice using classic methodologies. With Bichen, we made gene ESL targeting constructs to generate a STIN conditional knockout, as well as 2 STIN human allelic variants in mice. Bichen created the targeting constructs, Christina did the ESL targeting, Jimmy then did the blastocyst injection and created the chimers and bred them for transmission. So this is just an example for you of how we can use embryonic stem cells to create genetically modified mice for your research program. And it works very effectively, but it does take quite a bit of time. Another example I'd like to give you is creating a transgenic mouse. This was a mouse that we created for David Schwartz, Chris Evans, and Ivana Yang. They are interested in looking at Lyme mucins and airway defense, and they're particularly interested in MUC5AC and MUC5B because they represent the principal macromolecules in the airway. So they had a few tools on hand, and what they asked us to create for them was a MUC5B transgenic mouse. It was generated using BACs because this is a very large construct. It was over 35 kb and involved the MUC5B genomic DNA paired with a rat promoter to express in non-ciliated lung cells and a reporter, GFP, that enabled them to study mucociliary clearance and the role of MUC5B in that process. And as Molly discussed with you, CRISPR-Cas9 has really revolutionized how gene editing can happen. And for us particularly, it was of special interest because if we had the potential to create a double-stranded DNA break in a very specific area of interest to us in the genome, that created an opportunity for us to change that sequence. There are two components here, the CRISPR guide RNA, which has a 20 base pair recognition with target DNA. As you can see here, this is that orange hairpin. It will go and bind to the target DNA in the genome and recruit in the Cas9 nuclease, which will create a double-stranded DNA break. The most common outcome when that double-stranded DNA break occurs is non-homologous injoining, which is a random repair, and it can often cause frame shifts and lead to stop codons, which would create a null or knockout allele if that double-stranded DNA break occurred in a coding sequence. Another alternative approach to this, which we use, is to create safe knockouts, where instead of introducing one guide RNA to introduce a double-stranded DNA break, we would introduce two guides to cut in the same gene to remove intervening DNA and create a knockout. The other possibility is to use, to rely upon a more precise repair mechanism in the cells called homology-directed repair. And this requires both the guide RNA and Cas9 as well as a homology-directed repair template, which has some sequence similarity to the region that you want to repair, as well as a mutation or modification you want to introduce, which is illustrated here as this red box. And using this approach, we can introduce single nucleotide polymorphisms, we can introduce LOX-P sites to make conditional knockouts or reporters and tags. And in essence, what we do is very comparable to what you would do in a cell line, except in this instance, the cell is a single cell fertilized mouse egg. It's a zygote. So we would take these zygotes under the microscope, and we would inject the guide RNA, the Cas9 nuclease, and possibly a DNA template if we're looking for a precise repair. Those embryos are then transferred into mice, and the resulting pups or possible founders are interrogated to look for the desired genetic modification. An example of this for creating a safe knockout mice using CRISPR-Cas9 is just illustrated here. At the top, I have a very generic looking gene with introns and extrons. And we would design two guides that would cut out a very important piece of that DNA. Usually it's a functional region of the gene of interest. Those two guide RNAs would be co-injected with the Cas9 nuclease into mouse zygotes, transferred into pseudopregnant females, and the pups that were born would be interrogated for the deletion event by using primers outside that guide sequence, the intervening DNA that would have been removed by the guides. And here below is just a simple genotyping assay using those PCR primers, and you can see that each of these individual mice, some of them have a large PCR product and some have a smaller one, which would be indicative of a knockout. This is a process that takes 4 to 8 months, $4,000 to $6,000 to generate founders. Many times these founders can be homozygous, which means that in some instances, you could consider using those founders directly for analysis for preliminary data. So this is a very exciting way to generate some early data for a grant. One example of a safe knock that we made here is illustrated here. And we did this in collaboration with Kate Waugh and Joaquin Espinosa. Kate was working with this existing model of Down syndrome on the left here, DB16, which is basically contains these regions of about over 100 genes that are trisomic in this mouse. And those are illustrated by these blue boxes. When Kate came to me, she had a locus, a region of 4 genes in that trisomic region that she was particularly interested in because she was hypothesizing that those 4 genes were actually contributing heavily to the phenotypic Down syndrome-like features they were observing in the DP16 mice. And so what we did for her was we took a regular wild type mouse and we used 2 guide RNAs and Cas9 to create a safe knockout and remove about 200 kb of that sequence to remove those genes, that locus of interest that she was interested in, and create a knockout for that. And then she crossed those knockout mice with her DP16 mice and generated a new mouse, which many of you may have heard about, that she calls Norm, which is trisomic for all of the genes in DP16 except the genes that she's interested in evaluating for their contributing role to Down syndrome. Some other examples of knockout mice we've made, we have created founders in which all 4 alleles of a duplicated gene were disrupted, allowing for the investigator to generate some preliminary data for a grant proposal. We've created mice with varying size deletions up to 600 kb. We've created mice with a duplication event where that DNA was cut out, but it was also reinserted, so there was a duplication of a large piece of DNA. We've been able to delete putative enhancers and other regulatory elements for investigators, and potentially you could create knockout mice with more than one gene within a single project. The other win of this is precise repair following CRISPR-Cas9 double-stranded DNA breaks, and this is illustrated here where the most common request we get is for creating point mutations in mice. In this instance, as I told you before, you would use the CRISPR guide RNA in tandem with the Cas9 to create the double-stranded DNA break and introduce a homology-directed rare template. And this template would include DNA, as illustrated up here in this oligo, having homology with the genomic locus, but also introducing in there something in between there, something that they want to introduce, like a single nucleotide polymorphism. When we do this, we often introduce by silent mutation a novel restriction enzyme site to facilitate genotyping. So when pups are born, as you can see here at the bottom of the screen, each of these lanes is a different mouse. We do a PCR to amplify outside the region, around the region of interest, and we follow with digest with that novel restriction enzyme and look for evidence of homology-directed repair. And as you can see here, a good number of the mice, such as 3, 4, and 5, show evidence of homology-directed repair. So this is a quite robust event. An example of a mouse we made using this is from Beth Tamburini's lab at the university, making a PD-L1 cytoplasmic mutant mice. So Beth is particularly interested in the interface between antigen-presenting cells such as dendritic cells and T cells in immune response. And they engage with each other through a variety of different receptors, including MHC-presenting foreign antigen on occasion. And a lot of interest in this field is focused on what happens to that T cell when it engages with the antigen-presenting cell. What signals does it receive, and how does it make the decision what to do in response to its engagement with the dendritic cell? And Beth was interested in the dendritic cell and the possibility that there was some reverse signaling being transmitted into the dendritic cell following this engagement with the T cell. So she was particularly interested in the receptor PD-L1. And when she came to us, she was interested in changing a motif in the cytoplasmic tail, TSS, to 3 alanines, with the idea that she could test this hypothesis that this motif participated in reverse signaling in dendritic cells. So using the CRISPR-Cas9 and this homology-directed repair template, we were able to generate this mouse for her, and she was able to use that mouse in some studies and to show a functional difference in dendritic cell trafficking to draining lymph nodes following poly-IC injections. So this was a pretty exciting thing for her. Some other examples of mice we've made using homology-directed repair. The most popular request we've had is for using this approach to create single nucleotide polymorphisms that are associated in disease in human patients. We've done this for a number of investigators now. We can use this approach to create small tags rather easily, such as flag tags or MIC for genes of interest. We've created mice with mutations in splice acceptors or donors to preferentially support the production of certain isoforms of a gene. And we've created mice with mutations to putative binding sites for transcription factors for investigators to help them evaluate the role of that binding site and the expression of their gene. So most of the things I've been telling you about so far with the CRISPR-Cas9 are related to B6 mice. And there's been a historical reason for this. The B6 mice are the most widely used strain for biomedical research. They are the first fully annotated mouse genome. And most of the embryonic stem cell targeting that was done in B6 cells. So there was a kind of a funneling effect where the use of B6 mice as a background facilitated the use or the generation of more mice on the B6 background. But it's important to keep in mind that B6 mice have their own baseline phenotype, a susceptibility to diet-induced obesity, for example. And so while suitable for some models, it's not ideal for all models. And so we decided to take advantage of our capacity to do in vitro fertilization to enable us to rapidly make mice on different genetic backgrounds to help investigators get to where they wanted to go faster. So doing this, we've been able to use a variety of background strains, as they're listed below, to generate mouse embryos that we've used with CRISPR-Cas9 to directly generate mice with modifications on different backgrounds. So this is an example here is with the NOD mice, the non-obese diabetic mice. A lot of these have been done in collaboration with John Kapler at the Barbara Davis National Jewish, as well as Mark Anderson at UCSF. Using exome sequencing of type 1 diabetic patients and families, they've come to us with several candidate genes with SNP variants that they want to test. We've been able to make these modifications directly on the NOD background. It's enabled them to refine their knowledge of phenotype, the phenotypes they might see in early disease onset, shape their hypotheses, and now we are going to be making them mice using a panel of different background strains, a NOD congenic, and a B6, to help them further evaluate the role of these SNPs in disease in patients. Another example is with mice on the BALB/C background. These are very popular strain. This is a popular background strain for people working with Th2 biased immune responses. We had an investigator come to us that had backcrossed a reporter for a gene onto BALB/C for many, many generations, and he was interested in following some attack-seq analyses he'd done to assess whether or not a particular region upstream of that gene was acting as an enhancer for his gene of interest. And to help him with this, we used his mice to generate embryos for injection, and we used CRISPR-Cas9 to remove that putative enhancer, and he was able to quickly assess whether his gene of interest was impacted by the removal of that enhancer in the context of parasitic infection. And now we're going to move forward with that and continue to build on that mouse to remove different putative enhancers to create a reporter that's genetically linked with the initial reporter to help develop his studies further. In addition to that, we've done a lot of other mouse models with CRISPR, including multiple base pair changes over very large genes spanning over 30 kb. We're introducing modifications into already genetically modified mice. For example, if you had a Rosa 26 stop clocks mouse expressing your gene of interest, we could modify that gene of interest to introduce a SNP or another modification that would enable you to overexpress that modified variation of the gene. And we can create conditional knockout mice. The most challenging for us right now is to create mice using CRISPR-Cas9 with very large insertion or replacement events. This is just a quick depiction of some of the early results we've had through 2018, I believe, using CRISPR-Cas9. Each of these dots represents a different project, and the color of the dot is related to the background strain that was used. And there are two panels here. Basically, I'm plotting the percentage of modified pups that were born out of the total pups born from an injection project, either for knockouts, the percentage of mice that were knockouts, or the percentage of pups that were born that showed evidence of homology-directed repair. So you can see there's quite a wide range, but it so far has worked in all the instances that we've tried it. So I think it's a very powerful tool to enable you to very quickly address your questions on the background strain you want, making exactly the mouse you want. So this is in contrast to embryonic stem cell targeting. CRISPR-Cas9 can be much more in the area of months as opposed to years. So I'm almost done. I appreciate that we're running out of time. This is, the dogma of how we think about genes. Sometimes we think mostly about DNA transcribing into RNA, translating into protein, and depicted below are some of the tools that we've used to try to untangle the roles of different genes in our research programs. So classic transgenesis, the example of the MUC5B transgenic that we made, classic knockouts and knock-ins and conditional knockouts, such as the sting mice that we made for John Cambier. We can also make tetracycline inducible mice, which modulate transcription of a gene of interest. And as Molly reported as well, RNAi or shRNA can be used to directly modify RNA. And today I've been giving you some examples of how we've been using CRISPR-Cas9 in the germline, but we've also done it in somatic cells in the mouse to modify the DNA. And there are also a lot of tools coming up that will help you to further your research programs using CRISPR-Cas9 in unique ways. For transcription, there are Cas9s that are unable to create double-stranded DNA breaks, but they are affiliated or associated with activators or repressors of transcription, so they could be used to help you turn genes on or repress the expression of genes. And finally, the thing I'm probably most excited about are these RNA editing Cas proteins, such as Cas13, Cas711, and Cas-Rx, which conceivably would enable us to target gene editing to RNA as opposed to DNA. So if you were to try to reach us, you can go to the Research Corps' website on the Anschutz website that Vanessa has worked so hard to set up. You can also talk to people in the animal facility. We are in close contact with Jory, Mike, as well as the import-export coordinators. And they would be good people to reach out to. Or you can send an email to mousegenetics@njhealth.org. That does come into my mailbox, or email me directly. I'd like to say that I'd be very excited to speak with any of you, throw ideas around, even if you don't have a clear idea of what you want. I would be happy to speak with you about your projects. Okay, and with that, I'm going to stop and see if there's any questions.
Jen, I have a quick question for you.
Yep.
Something that you and I have talked about a little bit. If an allele that you are making is expected with CRISPR is expected to potentially be embryonic lethal, do you have, has the Corps been using methods that will maybe enrich for heterozygotes as opposed to getting homozygotes that might actually die before we could use them?
Yep. So there are two different ways to do that. The way that's been most successful for us is to create a second homology-directed repair template that is basically wild type, but has been mutated, so it's not recognized by the CRISPR-Cas9. So the hope would be that it would also be incorporated to some extent in the zygote and prevent us from having a CRISPR-Cas9 that's overexuberant and causes some kind of, knockout. The other possibility that we have done with some success, but it's a little bit more involved, is to inject two-cell stage embryos as opposed to the zygotes, the one-cell stage, where we are pretty confident by injecting one of those cells out of the two that we will have at least heterozygosity.
Okay, great. Thanks.
Yep. Okay, well, I think if there are no more questions, I think we're done for today. Thank you for coming. I appreciate you taking the time out of your day.
Thank you, everyone.
Thank you. That's great.
Thank you.
Bye.
Bye.